So today we have Rich Hite coming from Memorial Sloan Kettering Cancer Center. And Rich, what is the most robust part of the Cryo-EM workflow for you?
How much fun it is to see new protein structures.
Okay. Always a scientist. Okay. Rich, take it away.
Okay, thanks everyone and good morning. Okay, there's some feedback. Thanks everyone and good morning. Today I'm going to present on the interpretation and limitations of single particle analysis. The first half will be somewhat of an overview, and then I'll give two case studies from my lab as examples of how we try to handle cryo-EM data, what we can learn from it, and then also what we cannot learn from it, because it's not a super tool that can answer everything. It's not a silver bullet, but it can provide useful feedback. And so I think the first question that we all need to be commonly asking ourselves is, what is the value of an experimentally determined structure in the era of AlphaFold structure prediction? What are you getting out of going through all the work of expressing your protein, purifying your protein, putting it into a microscope, collecting data, processing the data? This is expensive time-wise, expensive resource-wise, expensive infrastructure-wise. And so what is it that you hope to get out of it? So can anybody answer it? Why are you here? What are you hoping to learn from using experimental biological structures of proteins or protein nucleic acid complexes?
To answer mechanistic questions.
Yeah. And I think that's fundamentally what it is AlphaFold can provide us with very pretty pictures. It can give us an idea of maybe domain organization, architecture, how things fold together, but it can't necessarily tell us mechanistic questions about how proteins work at a fundamental level. And for protein biochemists, that is in essence of what we're trying to do, is understand how proteins work, how they behave, how they respond to stimuli, so that we can understand how their functions lead to some physiology in a cellular or physiological context. And so based on that, we want to try to design our EM experiments to maximize the benefits of determining experimental structures. If your goal is just to determine a structure, AlphaFold may be sufficient. It may be able to give you sufficient information about how different domains fold together, where you have long-range and short-range interactions that you can perhaps test with an orthogonal approach. If you want to look at how stimuli influence conformation, how long-range allosteric mechanisms work, how the active site of an enzyme is actually able to catalyze its reaction, this is where AlphaFold cannot likely answer your question, and this is where cryo-EM can be particularly informative if you're able to design your experiment in a way to answer these questions. And so just a brief overview of cryo-EM. So I think you've probably heard about this a lot. So one of the great things is crystallization is not necessary. And one of the advantages of not needing crystals is you're not forced to just have one specific set of particles. You don't have to have just one conformation. You don't have to have just one composition. Sometimes that's great because it allows you to get high resolution. All of your particles look the same. But often biological specimens are dynamic. They have domains that move. They have active sites that open and close. They have substrate binding sites that you may not be able to fully saturate in your experimental conditions. And so by not needing a crystal where everything has to be identical in the entire lattice, you're able to sample this heterogeneity, sample this diversity, and this can provide significant information into how the protein may function, especially when we come back to the idea of what is the mechanism of the protein or protein complex that you're interested in looking at. And so going back to the beginning and how we've kind of made a progress to get to where we are with modern molecular cryo-EM, the first structure, does anybody recognize this protein here? Yes, it's the phage tail. And so this is, in my mind, one of my favorite papers and one that I revisit quite frequently because it's very clearly thought out, it's very clearly laid out, and it also provides a platform for which everything that we do is based on. And so in this case, it's a helical reconstruction of the bacteriophage T4 tail. And what they're able to do was find where the individual particles were, where the individual components of this helical filament were, and doing Fourier filtration, improve the signal to noise from that and generate the first 3D reconstruction. And from that, they're able to fall out how you can take two-dimensional projections and generate a three-dimensional reconstruction. But it was also obvious to the authors that most proteins are not helically symmetric, and you don't have all the views. But by forcing them to have different views just by tilting the stage, you're able to sample the same sort of space, of Fourier space, And so from this, you can generate three-dimensional reconstructions. And so I think this is a foundational paper. And for all of you who are interested in pursuing a career in cryo-EM, it's one that I would recommend taking the time to go back and revisit, because it's a simple paper at face value, but what was learned from it and what is available from it and the foundations that it have built are really quite impressive. And so thinking about how this works in cryo-EM is you have particles randomly floating in solution, unlike your helical reconstruction where you have all the views just by the tilt of the specimen along the helical axis. For normal, most common proteins, you don't have helical symmetry, and so when you put them in an aqueous solution, they're randomly floating in solution, and so when you pass the electron beam through them, you're able to get projection views that correspond to those different views of the sample. And so now you don't already have from your helical symmetry what are the orientations that are present in order to generate your reconstruction. You have to determine how all of these views correspond to one another. If you're able to do that, though, you're able to generate a three-dimensional reconstruction, whether this is from negative stain or from cryo-EM images. The theory is the same, is that you have some sort of orientation parameter that relates each one of your particles to your volume, and as you iterate that and get better and better alignment of your particles, you're able to get a better reconstruction of your volume. And so I think you've probably gone over this multiple times this week, but this is the foundation of how we're able to generate the reconstructions. And so, as I mentioned, there's negative stain where you have images of some sort of protein. You're not actually looking at the protein, but you're looking at what is presented around it using the negative stain, so usually some sort of heavy metal ion that wraps around the protein, and so you look at where the stain cannot access. This is very useful for small proteins or highly dynamic proteins where you're unlikely to get very stable averages. It can also be useful as a screening tool to see, do you have all the components of your complex? Because in a few minutes, you can prepare a negative stain grid, put it in the microscope, collect some images, and see, do you have a homogeneous composition, or do you have a whole lot of different sizes and shapes, indicating that your sample may not be ready to move on. And so there's a variety of uses for negative stain. Cryo is then when you, instead of looking at the stain around the protein, you're actually looking at the atoms of your protein, because those are what are scattering the electrons, and so they scatter more strongly than the aqueous solution around them, allowing you to visualize the protein structure at high detail. And so about 10 years ago or so, there was a foundational change in the way that cryo-EM was done. Prior to this, it was a few practitioners mostly looking at low resolution structures. Occasionally, you could get high resolution with a very small subset of proteins, but it was limited because of the technology itself. And I'm sure you guys have talked about this already, but this is a plot of dynamic quantum efficiency. And so this tells you approximately how many electrons you're going to be able to recover as they pass through the sample and hit the specimen detector. And so with film, which was state of the art up until this point, the best you could hope for was about 3 out of 10. And so due to the limitations of radiation damage, where most of the electrons that you're actually passing through your specimen are not beneficially, they're damaging your specimen rather than generating information, you get a very low yield. And so this limited the resolution, because with film, you couldn't recapture most of the electrons that were passing through. And then the real breakthrough was the development of these direct electron detectors. And so the blue curve corresponds to a previous generation, the K2 camera manufactured by Gatan. So I've actually looked a couple times, and no one has actually published a paper where they've quantified DQE in the same way as this report from a few years ago. And so I don't know exactly what the DQE is. Manufacturers have put them out, but I'd much rather have it from an experimental biologist where it could be benchmarked on their own system with their own sample. Have you seen something for some of the more modern cameras?
I know they have an MTF file, but I don't think anyone's in the IP on the sensor and did exactly this experiment.
Yeah, and so that's something every few years I go back and look for, but it hasn't been published in the way that this was. But the idea is that the new generation of cameras should be better, but the data isn't as readily available as this paper, even though this paper is now over 10 years old. This is a very nice demonstration of the quality of the K2 relative to film and other available detectors at this time. Any other questions?
I'm wondering what the x-axis actually means here. What does it mean to take fraction
So when you get to Nyquist, that's where you have no ability to fit your information because you can no longer fit a sine curve between 2 pixels. And so that is the maximum resolution of the camera. And so your specimen has a particular calculated pixel size because you've magnified your specimen to that particular camera. But the camera itself also has inherent technical resolution. And so this is, if you have, in this case, a K2, so 4K pixels or whatever it may be, 2K would be your fraction of Nyquist. And so then if you have a particular specimen and you have a magnification, you can figure out what that corresponds to in resolution for your particular protein of interest. And so for most specimen that we work in, this low resolution regime here, so between 0 and maybe 0.25, is actually much more important than you would generally think, because this is the low resolution terms, which are what align your protein overall. The high resolution are what you need to get the high resolution structures, but they're not as necessary for alignment. The alignment drives the reconstruction. If you have your particles poorly aligned, you're never going to be able to find those high resolution terms. And so even though there's only a small range where it has this very high DQE, that's what drives a lot of the reconstructions, because you're able to align your particles quite well. Then the high resolution terms fall out, although they're significantly dampened. And this is why we generally use a B factor to help weight up those high resolution terms. There's also more radiation damage of high resolution terms. So there's a convolution of the detector defects, your microscope potential movements that can't be corrected, as well as radiation damage. And those all get convoluted into the B factor to help weight up your high resolution terms. But the low resolution terms are essential to even get a chance to get those high resolution terms, because they're what drives the alignment of the particles. These are great questions, thank you. Any other questions? So that was, I think the question is, theoretically, you should be able to get to 100% efficiency, that every electron that hits the detector should be recorded. As I mentioned, there hasn't been an equivalent publication with the modern cameras, but it could be that at low resolution they're achieving that sort of regime. Or it could be that they're just getting better at higher frequency information. I don't know, but that would be something that perhaps you guys could convince Ed to have NYSBC or NCCAT invest the resources in actually benchmarking, because I think they probably have a full array of the cameras compared to just about anywhere else in the world. But the time and effort to do it is actually quite intensive. So it's a matter of what is the best use of resources. Okay, and so thinking about challenging specimen, if you've ever seen true atoms, you would assume that this is probably not a challenging specimen. This is the best that one could possibly do. These are density maps from a apoferritin structure that were published now several years ago, where they're able to get true atomic resolution from Cryo-EM. But most of us here will likely never reach that with any of the specimens that they actually care about, because of all of the issues that limit resolution. And so this can be movement of your stage, movement of the specimen, but because of the ability of the cameras and the microscope to achieve this, that shouldn't be the limitation. The limitation is generally more biological. Our specimen are dynamic, our specimen are noisy, they move, they're small, they're not necessarily have high symmetry. All of the things that help to allow apoferritin to achieve high resolution are generally not things that are present in the specimen that we work in, and that limits our ability to achieve high resolution. But that doesn't necessarily mean that we're not able to get more biological information. That dynamism, those movements, those conformational changes are actually what allow us to understand mechanism. And so there's an interplay between being able to get extremely high resolution and being able to answer biological questions. And so often when we start an experiment, we have a particular question in mind. Sometimes if you're looking at a true enzyme active site and you need to know precisely how the water is coordinated to facilitate some sort of attack, then you can get an idea of what the resolution is you need to be able to visualize that. Other times you're looking for what is the conformational landscape of your protein, and there high resolution may not even be your goal. You may be looking for hopefully high enough resolution where you can dock in atomic models and see how things are moving, but achieving sub 2 angstrom resolution in that case is probably not your goal, because in that case you want to get as many different conformations as possible to visualize the conformational landscape rather than trying to achieve high resolution. And so with each experiment that you go into, you need to think about what is the goal of the experiment, what is the question that you're trying to address, and based on that, then design the cryo-EM experiment to do that. If you have things like inhibitors, you can often improve the resolution because you're limiting conformational space. If you have things that are often sampling a broad range of conformational space, that's going to limit your ability to achieve high resolution, but it may help answer the biological question you have. And so there's an interplay between these two, and these are things that I'm going to come back to throughout the lecture, and things that I hope that even if nothing else of my lecture passes through, when you sit down and start an experiment, think about what it is you're trying to achieve, what it is you're hoping to learn, and what do you know about your sample? How can you potentially shift it to achieve that regime? And so when you first get your reconstruction at the end, one of the things that you need to do is evaluate it. We've spent now, if it's a new project, maybe 2 or 3 years doing the biochemistry, optimizing sample preparation, collecting data, determining the structure. Now you actually have your density map. What can you do with it? The first thing you need to do is visualize it. There's nothing that, looking at the FSC curve, looking at all the various validations from whatever your reconstruction algorithm are is useful, but the only way to truly evaluate your reconstruction is to open it up in some sort of visualization software, look at it and see what it's telling you. Because cryo-EM reconstructions are heterogeneous by nature. There's regions that are going to be well ordered, there's regions that are going to be poorly ordered, and that's information. That's information that you can use to help understand your system. And so I'm going to use an example of a drug-bound kinase complex that was published a few years ago as kind of a tutorial for how I like to open up a reconstruction and look at it. And so in this case, there's a multi-component complex, and they have this ligand shown here in purple that is, in this case, an inhibitor of the kinase. So when we look at the reconstruction, we can think about what does resolution mean? And so what does resolution mean to you guys? What is the definition of resolution in physics, if any of you are physicists? Yeah, how can you distinguish two different objects from one another, or do they become a single object? And so with cryo-EM, that corresponds to, can you separate two atoms, side chains, whatever it may be, and distinguish where they are? And so that also matters the spacing between objects. And so if you have a carbon-carbon bond, which has a distance of about 1.4 angstroms, you would need that sort of resolution that I presented at the beginning of the apoferritin structure to see individual carbon atoms. That's not something that we achieve very frequently, and in many cases isn't necessary because we often know the geometry of how proteins fold, and so we know if we can see where the carbon-carbon bond should be, we can often use geometry to model it. But other things, side chain rotaburs, there you often have different distances. Looking at waters, waters are generally resolved somewhere around 2.8 angstroms because that's often the distance between a water and a protein atom. And so if you're above 3 angstroms, you're unlikely to see waters. And so coming back to the idea of an enzyme active site, if you're below 3 angstroms, you're probably not going to be able to visualize the catalytic reaction because you're going to be missing critical features.
For example, if you're at 3 angstroms, then you see a very clear water density.
So I'm always conservative in the way that I do it because those benchmarks are based on your overall resolution, and you're interested in the individual features. And that's why I say use the benchmarks, use them as a way to understand what you're looking at, but then actually go through your density because your density map is the truest understanding of what you're seeing. The other issue with water is water doesn't look that dissimilar from sodium, which doesn't look that dissimilar from chloride. And so knowing what you actually have, you may see a very nice spherical density, but then you need to look at the local geometry. Is it something that could be potentially an ion, or is it definitely a water? And this can be very challenging because we don't have anomalous diffraction like with an X-ray experiment where you can say unambiguously, that's a bromine or that's a calcium. We don't have those tools. And so in some cases, you may have homologous structures, and that can inform where ion binding sites may be. Other times, you may need to do orthogonal experiments to validate, yes, that's an ion binding site, or that's more likely a water binding site. Any other questions? Okay, and so with the idea of resolution, we have, as was mentioned, there's often criterion. If you are trying to dock in a small molecule, you generally want 3 angstroms or better. If you're trying to dock in a water, 2.8 or so, but that varies depending on the resolution. The way that resolution is generated or calculated in a cryo-EM structure, there's 2 ways. There's the global resolution, where you just take your data set, you split it in 2 halves, independently calculate reconstructions, and then calculate Fourier shell correlations between those 2 half maps. And this gives you the overall number, and this is the one that you report in your table at the end of the paper. But the local resolution, although the numbers that are generated from all these local resolution tools aren't as important, the local resolution is often more important. And this should match with what you see by eye. And so there's a number of tools where you can calculate plots like this, where you color different parts of your protein based on what the estimated local resolution are. And I don't generally take these as fact. I take these as information that can guide you. The regions that are thought to be higher resolution are often where you can have more confidence in your build. And if you look and you actually see better features in that region, things like well-ordered spherical water molecules, that should be consistent with this. If it doesn't match up, then perhaps the local resolution software that you used wasn't used properly, or there's some sort of defect in your protein structure locally that you may need to return to your processing to address. But these can be helpful for guiding not just building globally, but regions of your protein that are particularly important in how you can build there. So things like water molecules, ions, ligands, you would hope that these are in the well-ordered regions of your protein. And so in this case, they were looking at an inhibitor, and you can see it's circled in this region here. And this is among the highest resolution regions of their protein, which helps with the confidence that they're able to build their inhibitor properly. But then you want to go back and actually look at that density. So if this is something that you're designing an experiment on, that particular inhibitor, and you've now made a 2nd generation inhibitor, or it's a new inhibitor and you don't know precisely how it's fitting, you want to go back and look very carefully how it's been docked. And so in this case, you can see that there's several rings, and you hope that the density fits there well, but then you also want to look at the local geometry. Because as I mentioned, we don't have tools to find site-specific labeling of small molecules or ions in our structures. And so you hope that the geometric relationship between your ligand matches the geometric relationship of your protein. You don't have very bad clashes with bonds. You don't have negative interactions between atoms in your protein and atoms in your ligand. And in this case, you can see they have a number of favorable interactions, and the distances are appropriate for these sorts of interactions. They also were able to see a biological meaning, that they were able to find the reason why this is selective for CDK2, but not for CDK7, where you have a specific clash. And so then they were able to make mutations, test that hypothesis, and that is a way to further validate their model.
The one on the right doesn't match, doesn't make sense, right? The ligand doesn't make sense.
Yeah. So this is where they were able to come up with a hypothesis for why, with the pink model, which is CDK2, you flip down this ring, whereas in CDK7 you now have a leucine, and that causes the ring to flip up. And that was how they're able to make a hypothesis for specificity. They could then make those mutations, change the affinity of their molecule for the drug, and this is a way to validate their hypothesis for why this was a selective inhibitor for CDK2, but was inactive against CDK7, or much less potent against CDK7. I think this was about 100 kilodaltons. I don't remember exactly, maybe a little smaller than that. This was a smaller complex, particularly at the time, 4 or 5 years ago when they reported this. This group has since gone on and done a whole slew of different inhibitors against this complex, and they were able to use the same sort of platform, and there they're able to achieve even higher resolution, which validated the findings from this first model, but showed that you can actually do small molecule drug discovery type projects with Cryo-EM. It's just very labor intensive because each project needs an entirely new structure from scratch, whereas with crystallography you can often soak in many molecules and in a few minutes shoot dozens of crystals and get a number of structures.
That's how accurate is the docking before density, like did they dock it correctly before the density is structure?
I don't think they did any docking prior. I think this was a molecule that came out of a screen. Once they had the density, then they could fit the model into the density in order to determine an experimental structure.
The stereochemistry of the carbon on the left was inferred from the synthesis, or that's
I assume it was inferred from the synthesis. Yeah, because trying to infer stereochemistry, even at 2.5 angstroms, you're most likely to get it wrong. That would be more where you'd need 1.5 angstroms or so, because the density is just not sufficient to be able to tell stereochemistry. And so this, again, if you don't know the stereochemistry of your molecule, your structure is going to have to be much higher resolution. Or if you have a racemic mixture and you don't know, is it binding both, is it binding one, these are things where you're going to need higher resolution. In this case, they just wanted to show that the drug could bind and they could get a reasonable pose, and so 2.5 angstroms was sufficient. And this is, again, going back to the idea I had at the beginning, that you need to make sure that the experiment you're designing can be achievable with the sample you have. And so this idea of resolution, and that, we often, yes?
I have a basic question to the previous slide. So if you are interested in just in this bucket, can you bias your collection of data to have more view of the active zone and to have a higher resolution locally?
So in some cases, in this molecule, it's essentially a rigid kinase, so there's not large local conformations. Sometimes something like, say, mTOR, which is another kinase but has huge local conformational differences, there you can use reconstruction approaches to zoom in on one specific region, and that may help your local resolution there. With smaller complexes, it's often challenging to zoom in on specific regions because you don't have enough variation elsewhere, and you don't have enough mass in one particular region to do a good focused refinement. But that is an approach that is often used for larger complexes. And so one of the things that I do every time I teach this lecture is I go back and look at the EMDB and try to see what fraction of structures are we actually achieving this better than 3 angstroms, better than 4 angstroms resolution, because we often think that every time we go and do a cryo-EM experiment, we're going to get high resolution, but that's certainly not the case. Even with modern technology, modern hardware, modern software, some samples are just very difficult to work with. And so when you design your experiment, think about this. And so we have somewhere around half or maybe better than 6 angstroms, but better than 6 angstroms is not atomic resolution. We only have maybe a third that are better than 4 angstroms. And so again, think about what the experiment is that you're designing. And so this was just pulled from the EMDB last week. I discarded the 2006 data because there wasn't much of it. But the idea is similar, and this trend is held, that although we get more and more structures, we still get a lot of, and a lot of these ones below 10 angstroms are probably tomographic reconstructions, which may bias it a little bit. But you see that we still have as many between 6 and 10, actually more between 4 and 10 as we do between 4 and above. So think about what the question is that you're trying to address and what sort of resolution you would need to be able to answer that question. In some cases, you need extremely high resolution, so you have to think very carefully how you can optimize your experiment to do that. In other cases, 3 to 4 angstroms may be sufficient. In some cases, 4 to 6 can be fully answer the biological question you're trying to address. And so design your experiment and design your data collection and processing based on what the biological question is that you're trying to address. And so I know you're going to talk more about model building tomorrow, but this used to be one of the huge bottlenecks. Is you'd get a cryo-EM density map, and if you're working with a large complex of several thousand amino acids, this could be a group effort of multiple people for several months in order to get an atomic structure. Now, if you have a reasonably high resolution structure, 3.5 angstroms or better, there's software tools where you can just give it the FASTA file, give it your density map, and in a couple hours it can spit back out a reasonably built first model of what your protein structure is. And in some cases they can even do nucleic acids. And so this is an example of Model Angelo, where they've built a neural network that can take your density map, take a FASTA file, and start building on its own. And over a few hours it can build large portions. And so we've had success, and I'll show you one of the complexes we worked on later, of where it was able to build thousands of amino acids in a few hours, whereas doing that by hand ourselves, it would have taken weeks to months, depending on how experienced someone is in model building.
So this is at 3.5 angstrom, but you build this for thousands of
Yeah.
How do you compare with Phoenix auto build?
We've had good success with this. Phoenix Auto Build, we've had a little less success. I think at higher resolutions, it doesn't make a difference. I think at this moderate resolution, Model Angelo can be particularly effective. But there's now a whole slew of other software, some of which are better in certain circumstances. And because they don't take very long to run, I would recommend that if you have a challenging model to build, just serially or in parallel run them all, look at the reconstructions, look at the models, and see how well you believe different portions of it. And then you can just cut and paste different pieces together to get what you would hope to be a reasonable starting model that you can then finish manually. And so this is now coming back to the idea I had earlier that I mentioned is that all EM density is the same, whether it's an ion, whether it's a water, whether it's a ligand, whether it's a protein side chain, and it can be difficult to distinguish them, even when you do achieve high resolution. So this is a structure we determined of an ion channel at about 2.5 angstrom resolution, and we had difficulty assigning the densities to ions or waters. And so in this case, what we did was we determined collected a second data set of a different ion that also binds in the ion binding site, but which scatters electrons more strongly because of its larger atomic number. And this gave us hypotheses as to which particular sites would correspond to ions and which particular sites would correspond to waters. And then we use mutagenesis to validate some of these binding sites. But the idea, again, is that Cryo-EM can be useful for telling you where things may be, but it doesn't necessarily tell you what those things precisely are. And so you need to take advantage of orthogonal experiments. And occasionally you can do a clever experiment like this, where you can use a different ion or a different substrate, and if they bind similarly, it can help you. But other cases, you don't necessarily have those tools, and so you have to use biophysics or binding assays, mutagenesis, to try to understand what you're actually looking at in your density map.
A lot of good stuff I want to ask. So McKinnon showed like the old density in the X-ray map. I think that was distinguishable. The X-ray map can tell the difference between the potassium and water at the time, isn't it? So did you, right? How did compare to the...
So when it was done initially with KCSA, they used anomalous diffraction, and they were able to identify precisely what atoms corresponded to ions, and they could also calculate the occupancy because they knew precisely what is the occupancy of the ions versus the occupancy of the local atoms in the protein. With EM, we don't have those tools, and so here we were not able to generate that sort of precision, and so we were able to identify sites that are most likely going to be ions, but there's no way to say that this is absolutely an ion, nor to say what is the occupancy of these sites.
So then you refine this map, the Cryo map, real space and not physical space, like just...
Yes, real space. Yeah. Yeah, and then we've used molecular dynamics, and those molecular dynamics simulations have supported the hypotheses here. They'll often see ions occupying the sites that were modeled as ions here in the molecular dynamics simulations. But again, these are all inferences. They're no absolute measurements.
So for example, if you want to find out the calcium binding domain, do you try with calcium, do you try without calcium, do you try with magnesium?
That's another way to try it. Particularly if you have something that undergoes a conformational change when calcium binds, that can be particularly informative that it is a calcium, because there you go minus and plus. This channel, we tried with sodium, we tried with potassium, we tried with cesium, we tried with several other ions, and the only ones that were that formed a meaningful structure were potassium and cesium. And so we weren't able to make those sort of inferences because there were no conformational changes associated with the ion occupancy of the pore. Okay, and so now I'd like to move on to heterogeneity, which is, to me, the most exciting part of Cryo-EM. And this paper that came out of the Ramakrishnan lab now about 10 years ago is to me, one of the foundational papers in Cryo-EM, the ability to really take a complex data set, parse it into different subsets, and learn new biology from it. And so in this case, they were trying to understand the structure of the mitochondrial ribosome. And at the time, they didn't have sufficient tools to do biochemistry such that they could exclusively purify mitochondrial ribosomes. They often had a mixture of mitochondrial ribosomes and cytosolic ribosomes, but they wanted to determine the structure of the mitochondrial ribosome. And so this was unsuitable for crystallography, but it was something that potentially EM could solve. And so they developed a classification strategy to try to look at just the structure of this mixed population that were the mitochondrial ribosomes. And so they used an approach where they started out with a full population, they generated an average reconstruction, and then with maximum likelihood, they were able to sort this into 6 populations, some of which corresponded to bad particles. A large number actually corresponded to cytosolic ribosomes, about 30%. But the remaining 65% were mitochondrial ribosomes, and even these were heterogeneous. They had some where they had intact small subunits, some where they had just a faint bit of a small subunit, and one where the small subunit was completely dissociated. And due to this, they didn't have sufficient signal to noise to do the full complex, but they could focus on the large subunit, which they knew from studies of bacterial and other eukaryotic ribosomes tends to be the most rigid portion of the complex. And so then they put a mask just around the large subunit, went through another round of classification. They were able to then get a population of about 47,000 particles. And from this, they could get a sufficiently high resolution reconstruction of the large subunit of the mitochondrial ribosome that they could determine a structure, even though they started out with a mixed population, not just mixtures of mitochondrial ribosomes, but it was also contaminated with cytosolic ribosomes. And so this really showed that Cryo-EM, using these maximum likelihood approaches, can sort out very heterogeneous data sets and identify distinct populations that you can learn biology about. Now, if you were able to go back, just because cameras are better, processing is better, from this same sort of population, you could probably determine structures of this entire ensemble of all of the different mitochondrial ribosomal states, as well as the cytosolic ribosomal state. But this showed that this was possible when computation was much more limiting and data collection was much slower. And so since then, there have been many advances in dealing with heterogeneity. And so one of them is not just having a simple one-step classification, but you can use masks and look at many different regions. And so this is another example from the LMB where they were looking at the spliceosome. And so they started out with this overall model of the spliceosome, and then through classification, they had about 150,000 particles at an intermediate resolution of the spliceosome. But they noticed that different regions had different local resolutions. And so here, because this is a large complex, they could start to use this idea of putting masks around different regions of the complex. And so they masked this region they called the head. They masked this region they called the foot. They masked this region they called the body. And from this, even though the overall numbers here are similar or perhaps worse in this overall 3.7 angstrom reconstruction, the local features of these were significantly improved compared to this consensus reconstruction, which enabled them to do a better job of modeling and visualizing what the structure looked like within this complex. And then they noticed that some of the regions, even with this single round of classification, were insufficiently resolved. So they used the two-step approach where they combined the arm, which is this little region here, with the body. First did one reconstruction of the arm and the body together with a focus refinement. Then they focused just in on the arm itself to try to do an even better job. And so this tiered hierarchical classification helped them improve the reconstruction. Then they also looked for global concerted movements, because here they were assuming that movement in the arm is not necessarily correlated with movement in the foot. And they wanted to see if there were correlated movements around the complex. And so here they did classification without using a mask, and they found two predominant conformations, one which had this open feature and one which had this closed feature, suggesting that there were correlated movements within the complex. To simplify this sort of tiered and separation classification, an algorithm was developed called multibody refinement, where you give it masks corresponding to different regions of the protein that you think move as rigid bodies, and then you can focus in on how they work. And in this case, to try to improve the signal to noise, you're not only just focusing in on the region that you're interested in, you're also eliminating the raw density for the other regions. So for example, here they have MRC. If they first want to look at the domain that they've labeled as M, they'll put a mask around M. Then they can multiply the volume, everything outside of that, by the inverse of it. So they can actually take the R and the C, which they've subtracted away, and now they have density that just corresponds to M in their raw particles. So this is actually manipulating the raw particles to try to improve the signal to noise. And you can do this iteratively for R and for C. Now you have three different reconstructions, one corresponding to the M domain, one corresponding to the R domain, and one corresponding to the C domain. And from this, you can then generate a new reconstruction of your particles where hopefully you have better resolution for each one of these regimes. And so this is an example, again, with the ribosome. So you can see these large movements, particularly in this head region, the lower resolution of the small subunit. And now with the multibody refinement, you can see that the resolution is significantly improved. So this is the most challenging part of this sort of tool, is getting your masks right. Often you have overlap between the masks, and so the regions at the interface are in both M and R, and that can help. But if you have too much of R in M, then you end up misaligning it because you're also incorporating the information from R. And so this is often an iterative process where you start with a first pass of just a rough mask, and then once you've done your multi-body refinement and see how these pieces actually move, then you can either tighten or expand your mask, or you can add domains, remove domains, and that can often be beneficial for improving the reconstructions. And because you know, in this case, how all of these correlate to one another, you can use eigenvectors and actually visualize the major degrees of movement. And so this movie here shows the movement along the first eigenvector, which is essentially the principal component of movement for this reconstruction. And you can see that there's a rotation of the small subunit, but then a much larger movement of the head domain. And this recapitulates work that had been done from crystal structures and other EM reconstructions of ribosomes. But here they're able to get a vast ensemble of conformations because they weren't forcing it into one state. They were looking at how the heterogeneity actually fet itself out in a single reconstruction. There's another tool, 3D variability analysis, which is part of CryoSpark, that uses a slightly different approach. Here, rather than saying mask around domain A, mask around domain B, mask around domain C, you're trying to look at the principal components of movement within a volume without knowing what this may be. And so in some cases, it may be useful to run this tool, 3D variability analysis, where again, you see the same sort of movements. This is a different ribosome, but the same sort of idea. You have this large movement of the head, more subtle movements of the other subunits. Here's the second principal component. And you can see now the small subunit is rotating and the head is moving. And this can then help you to identify where those masks might be, because this is a maskless procedure. But again, it gives you a similar sort of idea that you have this rotation of the small subunit and then a much larger rotation of the head domain. And this is, again, correlated with work that's been done on other ribosomal structures to see how they move during their catalytic reaction. And so here you can see the 4 different components, and they're all somewhat similar, indicating those are really the major movements that they're visualizing in this reconstruction. You can also use more complex approaches where you maybe try to put this into a latent space. So CryoDragon is an example of one where, rather than going through and just taking individual particles after extensive classification, here you can sometimes use this earlier and take the full landscape and try to plot it into some sort of latent 2-dimensional space like this. And then based on that, you can make hypotheses as to how a particle may traverse this to get an idea of what a reaction coordinate may be. And so this is, again, going to the spliceosome, and they're looking at different movements along a particular reaction coordinate, and then they can generate reconstructions of particles very local to those spots to get an idea of what the conformational changes might be of that reaction coordinate. And based on the density, you can see how likely it would be. So over here, where you have this large peak, but it's largely separated from the rest of the landscape, these may be distinct and will have a harder time traversing to the other states. Where the ones where you have a reasonably high density throughout, those may be continuous pathways in which the particle may move. Here's another example with a ribosome, but this is looking at how a ribosome is actually assembled. And so you can see different subunit assembly steps along the trajectory, and you can see how different features actually are added. And this is a way, rather than using classification, taking your full landscape to see perhaps how the ribosome in a stepwise fashion is assembled. And so these are all different ways to look at heterogeneity, and they can be very powerful. And often what we do in my lab is we try all of them and try them iteratively and try them sequentially to see what we can learn about the different conformations that are present in our data set to give us the best understanding of what we're actually visualizing when we collect our images. And so we have about 10 minutes left, so I'll just do one of the case studies. Yes? So this is not usually done with masks, no. And so this, you can do it you can do it in different ways. Sometimes you have such a complex landscape that it gets stuck just separating major state A from major state B. And so you may want to separate those out and then use something like Cryo-Dragon and look at all the substates. Other times they're sufficiently differentiated, like here, because you have large regions of the complex that are missing, it's able to well define each one of these populations in the latent space. And so this is what I mean, that we try different approaches, and we try them serially, and sometimes we try them sequentially, where you first try some sort of tiered hierarchical classification like I presented at the beginning, where you try to mix or separate subset A from subset B. Other times you may go directly to these latent space, find out what the different populations are, and then use those to perhaps do your classification again. So it depends on how you want to do it. Sometimes you may go through extensive pruning and really have a distinct subset of your data set. Other times you may just want to get rid of all of your false positives, and everything you believe to be a real particle you put in there. And then when you look at your latent space, if everything is all just one big circle, that suggests that you probably haven't been able to separate things very efficiently. But you can calculate reconstructions at different areas within that circle and see, are they significantly different? And then you can perhaps do a few rounds of classification to sort out if now, so you start out and you have 4 major conformations. If you separate into those 4 major conformations and then rerun Cryo-Dragon and you still just have circles, maybe Cryo-Dragon isn't the right tool for that, or maybe the sorts of heterogeneity it has is so subtle that it's not doing a great job with. And so in that case, one of the other approaches may be more beneficial.
And sorry, just one follow-up question. So once Cryo-Dragon is able to look at the data set, does it output sorted particles for the then take into CryoSpark?
So you can say in this case, I want all the particles that were binned in this group. So you color them B, all the ones C, all the ones E, and then you can put them under other tools. Alternatively, if you don't have something that's as well differentiated as this, like here, where you have this landscape, you maybe just say everything within a certain latent space distance of your blue dot, let's make a reconstruction. Everything within a certain distance of your latent space green dot, make a reconstruction. And so rather than trying to bin all of it together, you just go locally and look to see, are there conformational changes occurring? And that would give you more confidence in the analysis. Any other questions? Okay, so I'm going to go through quickly the first case study. I'll try, just because we're running low on time, I'll just quickly go through the biology. And so one of the questions that we try to address in the lab is how DNA replication works and how impediments to DNA replication can arrest DNA replication and can actually lead to replication stress and potentially cell death. And so the power of the replication is actually done by the CMG helicase, which is a hexameric helicase that unwinds double-stranded DNA to generate 2 single strands that can be catalyzed for replication by Pol epsilon on the leading strand and then Pol delta on the lagging strand with discontinuous synthesis of Okazaki fragments. And so this is a bidirectional complex. You move from a replication origin in either direction. And occasionally you can find impediments, something like R loops or G quadruplexes or RNA polymerases that can stall progression of this. And how do cells deal with this sort of replication stress? Because you don't want to have a partially replicated genome when you go into mitosis, because then you'll start tearing your genome into small bits. And so one of the complexes that we've been working on is how the replosome deals with things like G quadruplexes. And so using in vitro reconstitution, we can show that if you have a G quadruplex in the leading strand, you end up with these stalled right leading strands because it reaches that and it can no longer pass through. The helicase is insufficient to unwind these G quadruplexes. If it's on the lagging strand, it can pass through because it doesn't actually pass through the replosome. And so through biochemistry, we found that it was actually the helicase itself that was the impediment to stalling. And so we wanted to determine the structure. And so we purified the entire CMG complex. We had a template in which we had a G quadruplex or no G quadruplex, and then collected Cryo-EM images in the presence of ATP. And so here we were designing our experiment to visualize the full landscape of heterogeneity because this is the helicase itself is a AAA plus ATPase, and so it hydrolyzes ATP to generate the energy to unwind the DNA. And so rather than using a non-hydrolyzable analog that would lock it in one state, and that's often very useful for visualizing a state, we wanted to look at a number of states. And so here we gave it ATP and hoped that it would unwind, and then similar to our in vitro biochemistry, it would actually stall at the G quadruplex. And so we could visualize precisely what it would look like when it was stalling.
So if you have a ATP analog or ADP bound form, like would that stall the complex?
It will assemble at the fork, but it won't progress to the G quadruplex.
I mean, it assembles the CMG.
It'll assemble the CNG on the fork, and people have done many structures of that, but it won't progress. If you want it to progress, then you have to add hydrolyzable ATP.
The known structure out there is that's just ADP now only.
So most people have done it with ATP gamma S or ADP AMP PNP, and there it assembles, but it doesn't progress.
Is there any structure with ADP now?
No, ADP has a low affinity and often dissociates from the complex. It's a purified complex.
So from cells down here?
No, so we purify individual subcomplexes. So we purified MCM 2 through 7, which is the hexameric ring. We purified CMG, and then we assemble that in vitro. And so here were this is a representative image and some of the 2D averages, and you can see we're able to achieve different views. Some of the densities, for example, here you can see this is much fuzzier than the density here, indicating that there's heterogeneity present in the data set. And so we're able to get a consensus reconstruction at about 3 angstroms. And so to try to visualize what this consensus reconstruction is actually composed of, what different states did we have present, we turn to 3D variability analysis, and I hope you can appreciate that there's large regions of the density that are just disappearing as we move through, and I think this was the major principal component. This whole region of the complex is disappearing along this principal component. And so this suggested that maybe our first thing should be a classification where we separate using a mask, either focusing on this region or another region that's correlated. So we first tried a mask around this region and tried to separate our particles. And so this corresponds to that mask around this domain. And we had about a 50-50 split. Half the particles had strong density in this region, and half the particles had no density in this region. And this corresponds to the fork protection complex, which is known to disassociate from CMG in particular replication states. And so this made sense that these are two components, these are two polypeptides that can bind and unbind from the complex. And so that was our first principal component. The next principal component we found is even when the fork protection complex was there, we had this large movement of this domain. It seemed to be almost a ratcheting. And being as this is a AAA plus ATPase that often undergoes ratcheting movements, and that's how they are able to propagate ATP hydrolysis to some sort of functional output, we thought that this might correspond to part of the actual activity of the helicase unwinding. And so we used now a focus classification on the C tier and separated the data set into many different states. Here, though, we have states. We don't know what these functionally correspond to. And this is often the biggest challenge that we have in interpreting cryo-EM structures is you get state 1, state 2, state 3, but we have some sort of in vitro experiment where we think this might correspond to maybe unwound, this may correspond to something else. We're thinking about an ion channel. Is this an open state, a closed state, something intermediate? And trying to assign discrete functional states to cryo-EM structures is challenging. And so we were able to reconstruct some of these, and using focus classification refinement, we got higher resolution reconstructions. But now we wanted to identify what these states corresponded to. And so this goes now, you have your density, go and look at it. It's your best tool for understanding what you're looking at. And so we're able to build atomic models in this. This is a structure we used the tool Model Angelo to build most of the model. It worked very efficiently. Within a few hours, we had, I think, 85% of the structure built, and then it just took us another week or so to build the rest of it. And then we could start to align it. We could see double-stranded DNA, we could see the fork protection complex, we could see the MCM complex, and we could see the CDC45 GINS complex as well. We then looked within the center of the complex, and now we could see a feature that we thought corresponded to our G quadruplex. But we noticed that the G quadruplex was in two distinct states. And so we knew this was the impediment that was actually stalling our DNA unwinding. And so we were particularly interested in what these states might correspond to. And when we looked, we noticed that there were a number of helical hairpins that are present within the structure, and these are actually thought to be what grabs the DNA and pulls it through during the ATP hydrolysis cycle, and this is what catalyzes unwinding. But in our case, due to the presence of the G quadruplex, it couldn't grab onto the DNA in the same way. It was grabbing onto this face that couldn't be pulled through the complex, and so this is our hypothesis for how it's stalling, is that rather than having a single-stranded piece of DNA that it can pull through a narrow constriction, now it has this large DNA secondary structure element that can't fit through as it's grabbing and trying to pull. We then compared this with existing structures as well as our structures, and what we found was that for existing structures there were two predominant conformations. There was a planar configuration of these hairpins as they're grabbing DNA, and then there was a spiral configuration. And what this effectively did is it changed the orientation of the DNA with respect to the complex. And then we looked in our states, we found one that very closely matched the planar state, but in the second state, now we had something intermediate between this planar and spiral, suggesting that it's some sort of movement of these hairpins that actually propagate. And that gave us information as to how it was able to unwind the complex. And so this is a model for how we think it occurs, is that first MCM2 reaches up, grabs the DNA, then MCM5 will reach up and grab the next part, and as it's passed through, right above this, there's a specific side chain that separates the two the two strands of DNA, what's known as the separation pin, and this causes the lagging strand to be expelled outside of the complex, the leading strand to be passed through. And through this serial analysis, or through this serial mechanism, you can unwind a long strand of double-stranded DNA, and it's continuous. As long as you have ATP, you can grab, pull, unwind, grab, pull, unwind. Do you have a question?
You have a diamond, or diamond sitting on the DMG complex the whole time. Like you just showed to the left is sitting on all the confirmations. Right?
Mm-hmm.
So an ATP, you call ATP in the point.
We can see ATP in 4 of the subunits in one of the states and 5 in the other state.
So it's not always all ATP sitting in 6.
No, because ATP can be, and we know from ATPase assays, it's actually being hydrolyzed in our reaction. And so this is an active enzyme, but because of the G quadruplex, it's not able to move forward. It keeps hydrolyzing ATP to try to pull, but it's not able to productively hydrolyze it.
So for a normal reaction, so if you have a normal, without
it would just pass through, it would unwind the template, and it would fall off.
You cannot track that, right? Because it just turn over so high that it
Yes.
fall off.
Yeah, and that's why previously people have had to use non-hydrolyzable analogs, and it's been limited in capturing distinct states because you can only assemble it at a fork and then add the non-hydrolyzable analog, whereas here we're able to move beyond the fork and capture additional states. And so using the same sort of approach, we've now modified our template in a variety of ways, and we've captured 6 or 7 additional states, and so we're hoping to be able to get the full reaction coordinate, and that's the follow-up that work.
I was just wondering, were you able to capture like the conformational changes based on the hydrolyzing of ATP?
So this was just 2 states, and I mentioned we did see a correlated movement depending on where the ATP was, but now we've gone up, and by changing the template, we can get different states along that, because the physical interaction between the DNA and the complex limits the heterogeneity that we can observe. And so by changing the distance between the G quadruplex and the fork, we can rearrange that, and then we can now correlate what ATP hydrolysis is doing to the actual conformational changes within the complex.
There's various states, and that would be the other way kind of suggested to show that you get reaction coordinate. Have you tried cryo-dragon?
We haven't tried Cryo-Dragon with this. Cryo-Dragon is technically challenging still, which we've had some people that have had success with it, other people that haven't. And here with 3D variability, we were very nicely able to separate it out, and it fit with the biology. And so we often start with 3D variability, 1, because it's very fast, and 2, because it's often easy to interpret because you're looking at movies. Cryo-Dragon can be more complex because latent space, you then have to make many, many reconstructions within that latent space and try to generate hypotheses for what they're showing you. But it can be more informative for much more complex things.
Do you usually take the particles from the 3D variability job? That was how they actually let you get particles based on the components?
We do it both ways. So what often is most effective for us is we'll take, if say you have 20 steps along your 3D variability component, we'll take maybe the particles in the first 3 and the last 3, generate reconstructions from those, and then use those as the inputs for a focused 3D classification. So for example, here, we tried it that way on this particular domain that was disappearing, and that worked okay, but it actually worked better just to put a mask there because we knew precisely where we wanted to focus our classification on. For other complexes, it has been more effective to take the endpoints of the 3D variability, which should be the 2 extremes, and then use those as the seeds for classification, and then it will move the particles that belong there, because the 3D variability isn't really a classification tool, but it can give you reconstructions that are informative. It depends on your system. Sometimes we've been able to mask maybe a 5 kilodalton domain. Other times we've needed 40 kilodaltons to be able to get an effective mask. And so we often try different masks, different sizes, and see what works effectively. Sometimes it depends on the signal to noise of your system. Sometimes it depends on occupancy. And I don't have a hard rule, and so I often tell people to try a couple things and then look at the results and try to see if you can come up with any conclusions. Okay, so I think I'll just skip the second one, and I can share this with anyone who's interested. But
One last question before you move on to the so if you compare the structure of the star complex with previous published structure, do you see the difference in the ATP binding pocket? Like ADP, I mean, at a different confirmation where we get ADP falls off, but you said it's not low affinity. So that means the confirmation has to be very different, right, between the ADP bound and the star complex. That is different.
So,
the ATP bound.
So the difficulty is, in the previous ones, they had non or slowly hydrolyzable nucleotide analogs. So they could see nucleotide there and they assumed that it was ATP. But there, if you do an actual assay, you see that there is turnover of slow nucleotide analogs. So whether it's precisely an ATP gamma S or perhaps ADP, they don't know from their assays. And so it makes it difficult to compare ours with theirs. The other issue is that some of the ATPase sites are very dynamic, and so we can't determine, we know that there's a nucleotide there, but we're not precisely able to determine whether it's ATP or ADP because it's so dynamic in some of the states.
But the one that's stable, right, that had analog in there, do look very similar to the pocket from your...
The pockets are the same.
They look in space.
Yeah, so the organization of the pocket's the same, but it can be difficult to assign, and in some cases it looks like maybe it's a mixed occupancy of ATP and ADP.
But if you put anything that will fall off, that means that...
It doesn't, it releases slowly, but we set up our reaction over 30 minutes, and so because it's a turnover reaction, it will release over time, but it does have some tendency to persist.
But would you think that ADP form will be different, the inactive form will be different from the active form?
Yes, yeah, and so we can see that the active site opens up in the ADP form, whereas it's closed with ATP because you have a residue from the adjacent subunit that's part of the catalytic site. Depending on what you're trying to do with it. So if you want to classify, then you can use a very tight mask just around the region of interest. So sometimes, even if you have a ligand, you can do a mask just around the ligand. It may be, in that case, 1 or 2 kilodalton plus some side chains, and that can be informative. Other times, you may be looking for large conformational chains. Maybe you think you have a continuous movement of a domain, and then you have a very generous mask around it, and there you're trying to see, can you classify those out? Excuse me. Alternatively, you may be trying to get a better reconstruction. So rather than using a classification for your analysis, you take all of your particles and you focus in just on one region. And in our case, we often do both. where we know that locally we have small movements, but we have much larger movements globally. So we first try to sort out the global movements, and then using refinement with masks, we try to improve the local reconstructions. Any other questions? Okay, thank you, everyone.